So I'm John Bell. I've been here at UCSD for the past 14 years. I'm a professor of medicine, Division of Hospital Medicine, and now the Associate Chief Medical Informatics Officer for inpatient here. And I'm going to talk to you today about the ENACT CCES, or the Clinical Concierge Evidence Service, which is how we're naming our new service. This is basically the goal of this is to take some real world evidence and to assist clinicians with some real world questions. Um, so the next. So here's what we're going to be doing today. So I'll take you through kind of and just with an example to show you some of the data gap that our current clinicians experience and potentially why real world data can help bridge that gap. And then we'll kind of talk a little bit about just the overall structure of the ENACT Clinical Concierge Evidence Service. And then we'll go through the workflow that the Clinical Concierge Evidence Service uses currently to take clinical questions, make them executable queries, and then get back understandable reports to clinicians. We'll then go through a couple different examples of that we've actually done, taking us through the process and kind of show you what we've the output of that. And then we'll take you through some of the limitations and the current challenges we're experiencing, and also then look at how we're trying to overcome those current limitations and challenges, as well as some of the roadmap for the future and some of the next steps that we're going to be taking with this service that we've developed. So first of all, one of the things that just had us develop the service in the first place was the fact that there's clinical questions from clinicians that they have a hard time getting good answers to right now. They often lack timely patient-specific evidence at the point of care. There's new situations that a clinician will encounter that they haven't experienced before and don't have great evidence for to pull from their back pocket. Or there's challenges to proceeding down a more typical path. So I'll give an example from a patient I experienced actually about a week ago for this. Is I had a patient, I was on the inpatient hospital medicine service, was taking care of a patient who came in with cholangitis. They were found to have an E. coli bacteremia. They went in and had ERCP, had their bile ducts open up with the ERCP, got a little bit of sludge out. They were initially put on empiric Zosyn or piperacillin and tazobactam, and then it came time that they were feeling better about day 3 of their hospitalization. We've gotten back the sensitivities to their E. coli in their blood, and are trying to think about how do we discharge this patient. And I need to figure out what am I going to give them to finish out the rest of their clinical course. I took a look at the sensitivities on that E. coli culture and saw that I had two kind of options. It was sensitive to Cipro and it was sensitive to ceftriaxone, which I know has some oral options, kind of third generation cephalosporins. It was the E. coli was Bactrim resistant. And so those were kind of my two good options to think, or my two options I was thinking about. I went and looked at that specific patient, and I probably would have chosen Cipro, had been any patient, but this patient happened to have an allergy to Cipro. So I was limited about what I could do there. And so I was like, how does cefpodoxime do as bacteria, as for coverage for finishing up a 7-day a 7-day course of antibiotics as a PO option for patients with E. coli bacteremia that has arisen from cholangitis. So I kind of had that question, I'm like, well, how do I figure out the answer to that? So let me kind of take you through the next few steps. So one of the first things is I can go and look at peer-reviewed literature. So right now, the peer-reviewed literature provides good scientific rigor. I usually feel pretty good about using it when it's available. But there's a couple challenges that I experience with it, and probably a lot of other clinicians do too. One is, can you find the right study quickly? And then two, did the results apply to my patient? So I looked, through try to do some literature searches on this example. I found some examples of people using what antibody can you use to finish up an oral course for a potential E. coli bacteremia. But one of my issues is that the patients in those studies mainly had E. coli that originated from the urine, and so I couldn't find any good studies looking at patients that had an E. coli coming from cholangitis. So then I'm like, I also, the other thing that we like to use, clinical guidelines are great when we have them. One, these also take literature. They've also had some organizational vetting of the literature. So you might have either a nonprofit organization or a physician's organization will take a look at the literature and say, here's what to do in this situation for this type of patient. And they're widely applicable to patients because they kind of cover big topics that we see, commonly, but there are many topics that aren't covered by them. So is one of the main issues with them. There's a great when we have them, um, especially if you don't have any contradicting clinical guidelines, which happens occasionally when you have two different organizations covering the same thing. But there's many areas that we don't have them in, and so we don't have them in. How do you? And on my topic, I couldn't find any clinical guidelines that said what to do. So then the newest kind of things out there are looking at expert opinion overlaid on top of clinical evidence or AI summarization. So there's tools such as UpToDate or Dynamed, which provide a layer of expert opinion upon the published literature. But again, these are fairly comprehensive, but they don't cover all topics. So I looked up this topic in particular and saw the main thing that it covered was should be an appropriate empiric antibiotic regimen. It didn't say anything about what to do, how to finish up an antibiotic regimen with an oral once you had sensitivities and once you had those bile ducts opened. So it didn't really help me with my clinical question. And then secondly, I said, I can use AI summarization. So open evidence is kind of the tool that everybody's using now for this particularly. It summarizes published data that's out there, but is also prone to hallucination because it wants to be helpful. So it wants to take your question and get you that last mile and get you that answer. So I actually put this question in open evidence and looked at it and it said, hey, yeah, I think that this would be a reasonable choice to do. And it gave me some explanation, but again, it was relying just on the studies that I found earlier in my peer review literature search that showed it was all based upon observational studies that were looked at E. coli originating from the urine and not from patients with cholangitis. And so again, it's like, is this really going to answer my question? I don't know if I can really trust this because this again is taking kind of what I was looking for and getting me the last mile without like really the evidence that I was looking at, hoping it would provide me. So if in a situation where we don't have, with the current options there's this data gap that exists. And so how can we address this data gap? So there's the potential of real world data. So you have this real world data that's out there in everybody's EMR, and it says, rich, has a lot of different information on routine care. So it covers all these things where the clinicians are having to make decisions on. It's, widely applicable to a bunch of different clinical situations as well. However, there's some challenges to this. One, the data sources aren't widely accessible. You have to have certain access to these databases to be able to access the real world data. Not just every clinician can do it. And then also often you have to require specialized training to mine answers for clinical questions. So one example probably of a fairly accessible real world data source is that a lot of people who have Epic is many institutions have allowed to use Slicer Dicer. But looking at our institution, which has been very liberal in its ability to, and what is given access to for the clinicians is basically given all clinicians access to that database. Is one, you see the utilization of it is there's been a few super users who use it all the time, and there's many clinicians who don't even have never even accessed it, even though it's been available to them for many, many, many years. Secondly, it takes so a lot of know-how to kind of figure out how do I get my clinical data to answer my clinical question from the data source. And so even when you have sometimes it being accessible, it requires more specialized training for those to be able to get answers to their clinical questions. So that kind of brought us to like this idea of, hey, we have the ENACT network. So let's create a clinical concierge evidence service that may be able to help provide clinicians with some real world data. And we also have a team that has some expertise to be able to extract that data from the real world data source that we have with the ENACT network. So that's kind of what brought us to creating the ENACT CCES. So looking at this, some advantages potentially that the CCS approach has versus traditional research is one, traditional research, there's a, even if you come up with an answer to a clinical question, there's a significant publication lag. So you finished your research, you've written it up, now you got to get it published to get it out there to people, to those clinicians who might be able to utilize it. So that's going to, it often takes months to potentially years to get out there. Many times the research is single site or registry limited because it's more expensive to do it with multi-site studies. And then also it can be expensive and resource intensive. And then finally, it takes a long time to get it out there to the clinicians. Everybody has heard those stories saying that, hey, there's been new evidence showing that this is the best new antihypertensive drug. And how long does it take to get to the clinicians? It takes 10 years for them to adopt. So there's a long time for it becoming bench to the bedside. So with our CCES approach, one, we can potentially get results with a quick turnaround. We have a greater than 50 site federated network, so we have multi-site studies or evidence from multi-sites using this structure. We're utilizing current EHR infrastructure and the data that's in there, so it doesn't cost anything to get that data from what we have. And then also we create reports that are designed for the direct clinician use. So it's it's not written for just for publication. We have the reports that are written to help that clinician be able to understand the data they're getting back. But again, I just want to emphasize that while we have potentially the way to get clinicians an answer to some of their questions or some data for some of their questions in a quick way, it is not meant to replace prospective research. So this is, basically to complement it and not to replace it. So for those of you who are still out there looking to publish your research, we're not looking to take your job. So feel safe. So again, the ENACT Clinical Concierge Evidence Service, our purpose is one, we're looking to have some on demand. So when clinicians have an actual question, be able to give them some real world data that helps with their patient care. The basic workflow, which we'll go into more details in a little bit, the clinician will submit a question. The CCES team will formulate queries to be able to then put it into the Shrine portal to the ENACT network. Then we'll take the counts that we get from Shrine, have a statistics team take a look at them and give a statistical analysis, and then we'll generate a report that we send back to the clinician. And again, the goal for this is to have timely data-driven support. Again, not prescriptive recommendations. So we're giving clinicians evidence so they can take back to the bedside and back to their office to say, hey, this is going to help me not rely just on my clinical experience, but I now have some more evidence in this for this clinical question I have. And this report doesn't tell me what to do. And so I can go back and make that decision with the patient. And so again, the reports are for clinical decision making, and then they also are really designed so the clinician can go back to the patient and be able to have a good conversation with shared decision making. So previously, to the past couple of years, patients would come. They have a question. Hey, doc, I've seen this new drug that I've heard about on all these commercials that I see, and I was watching my TV last night. Will this drug benefit me? And and previously, or they'd go and say, hey, I've asked Dr. Google, is this new GLP-1 gonna help me keep my leg? And the clinicians would have to come back and say, well, yeah, here's what we have. Here's the data that we have from research studies. But this is be able to enable us to be able to say, hey, we've got here's some evidence to say this may or may not help you, and here we can decide whether this would be a good decision or good next step for you in your clinical situation. Because now we're having patients not only coming with Dr. Google, but we're having them coming with Dr. Claude and Dr. ChatGPT as well. So they're kind of getting out there, getting more savvy about what evidence is out there, and it's a way for clinicians to be able to quickly get some evidence themselves to help them be able to provide appropriate evidence to go over these clinical questions with patients. So again, what the CCS is it's a place for them to get clinical questions addressed, to utilize a standardized workflow, It aggregates multi-site data for clinicians, and it gives them that evidence found to aid the shared decision-making with patients. What it is not, it doesn't replace randomized controlled trials or prospective studies. It complements, doesn't supplant that trial evidence. It's not a patient level data warehouse. You can't go there and ask for like, hey, I want to find a bunch of patients like mine and be able to then go and see what the people have done on those exact patients. It gives us aggregate counts and not in a no PHI leaves any of the sites at any point. And again, it's not a service that tells clinicians what to do. It just helps them provide evidence to help them in their decision-making process. So what is the CCS value proposition for clinicians, patients, and institutions? So for clinicians, the value proposition is that it will allow for rapid, tailored evidence when literature is lacking. So when you don't have literature that has or other ways to get evidence on a clinical question, it gives you a way to get some evidence for those questions. For patients, it helps for better informed shared decision making. So instead of just relying on your doctor's clinical experience, you're able to actually potentially bring the doctors are also able to bring some evidence to help them help explain how they're coming to their conclusions about what to do next, and share that with the patient. So the patient is also able to help with that shared decision making, and not just based upon what they've heard on from Dr. Google or from the TV commercials. As well as for institutions, they can feel comfortable about this because it leverages the multi-centered study or federated data, but there's no centralizing of PHI, so they don't have to worry about any of their PHI from their EHRs from leaking out and getting out there and causing HIPAA incidents. So, all right, so let's get into the a little more details about what the workflow for the Clinical Concierge Evidence Service is. So one, we first take the question and have to define that question from the analyst. And so oftentimes that times takes a little bit of going back and forth, because you may have a clinician who has a question, but is not able to put it in terms initially that allow us to develop queries for it. And so it may take some back and forth initially to help define that question. So there was a question one time that asked for what happened to patients who were put on JAK inhibitors who had a serious infection. And so we had to figure out, well, what do you mean by serious infection? Because that's kind of an example of how we needed to define the clinical question to make sure that when we got back to that clinician, we'll be able to answer the question appropriately. And then once we have defined the clinical question, we encode it as a Shrine compatible query. So we got to take that question and be able to break it down into the parts and figure out how you create queries that we can put into Shrine. Then we take we put those queries into Shrine, and we get federated site counts back. And then once we have those counts back, we send those counts to our statistical group and analyze the counts and give us an initial report with that analysis. And then we'll take that and then generate a clinician report. So I'll have a structured one-page summary, typically, sometimes 2 pages if you have a forest plot, and be able to have clinicians look at that to make sure it is in clinician-ready language to be able to deliver to the clinician. All right, and so here's a flow diagram kind of showing all of that. You don't have to be able to read all of this here. We're going to break it down into each of those 5 portions here in just a second. So don't worry about getting all the data here where you'll see it again. But here's the overall flow diagram of how we take it from the initial intake request to the final report. So in that first step, where we're defining the clinical question, so we take the non CCES clinician who submits the clinical question. We, as I said, go back and forth some on that to figure out, hey, we need to define the parameters that you were asking about to make sure that we have a good structured a question that we can then figure out how to make queries that we can put into Shrine. Then the other thing we'll do is we'll do a brief lit review to ensure that this question is not already answered. So if you come to us and saying, hey, what is an appropriate initial antihypertensive to give to my patient in general, we'll say, hey, there's a trial out there that already looks at that. We'll say you can go look at the All Hat trial just to say maybe that should give you your answer and not us. And so we'll do a brief lit search. If there's some literature that directly answers the question, we'll get back to the clinician and say, here's what you should maybe looking at instead of looking at this real world data. Then once and so as they're developing the question, we are trying to get most of our clinicians to create questions in a PICO format, where we get a population, figuring out which patients this might apply to, what's the intervention, like what drug or device or procedure are you using to want to see how it affects the patients. What should be the comparator? Is it another drug or device? Is it against the standard care? And so then figure out what outcome are they looking at to figure out which one is going to be the patient-centered outcome that they're looking at, if hopefully. So that's what we're using. And as that, we're beginning to think about some of the coding standards for each of those areas. And that kind of is the bridge that it takes us into our next portion of defining the clinical queries. So we're going to, as we define these queries, we have our CCS clinicians and our data analyst are kind of who is doing that now. We've talked about using some large language models initially to do some of that work, just putting it into some publicly available large language models to generate some of that work. And we've done some of that on some of them, but a lot of times we're still using our clinicians and analysts to think through that query and come up with how we would define that. Because the reason for that is a lot of the large language models that are out there, we can't put the ontology from Shrine into them easily. And so we don't always get the best output from them. So what we have done is our CCS clinician defines the clinical variables from the clinical question. And once we have those clinical variables, we then map those concepts to the ontology from the Shrine database. So we'll figure out what are the ICD-10 codes for those diagnoses. What are the LOINC codes for the lab value? What are the and so we use that to define all our variables. And then we have to come up with a series of inclusion and exclusion criteria, including the with, without, and when logic within Shrine, to be able to figure out how are we going to be able to put these clinical variables into Shrine. So we got to figure out, we've got a patient with who has cholangitis and has that E. coli bacteremia, and then we're figuring out, well, we want to look at exclude patients who are then treated with Bactrim or with Augmentin, but then look at patients who have Cipro versus cefodoxime. And then so we're looking at patients, maybe then using that when logic to say patients who have that the cholangitis and then are treated with one of those two antibiotics, or when do they have a subsequent hospitalization for cholangitis again in the next month? And so you're trying to figure out what might be your outcome. And then we come up with a naming search for our ACT network. So basically, we come up with a naming criteria to figure out how we put these different queries into the Shrine to be able to figure out what we're asking with each query. And then we create a Word document with all that information. And we've also often sent that to our statistical team so they can take a quick look at this, make sure that how we're going to these group of queries that we've developed is going to be something that they're going to be able to analyze appropriately with their statistical modeling. So again, we're taking that I2B2 ontology mapping, the ICD-10s, the RX norms, and the LOINC and CPT codes to take those clinical entities and put them into searchable entities that we can put into Shrine. We're looking for that inclusion and exclusion criteria as we're building the queries, and how do we build them utilizing the available logic that's within Shrine. And then we're looking for this, and including that, we're looking for the temporal relationships. We have to be careful with those temporal relationships. We only have a certain number of sequential logic events in Shrine, and we also have sometimes have had to use some date limitations around some of our query variables to figure out how to make sure that this happens before that when we have run out of the event logic in Shrine. And then we'll look at the statistical checks. Like I said, we'll send our initial queries to the statistical team to make sure that it lines up with their statistical models that they are able to run on the queries and on the counts that we'll get back. So then next we take the federated we take the queries, put them into Shrine, and get our actual counts from the federated database that is ENACT, or federated network that is ENACT. So the data analyst will put the clinical queries into Shrine. They will extract, transform, and load that, and then take all the counts, put them in Excel files, and format them in a standardized way to give to our statistical team. And then they'll have a definitions tab that goes along with those Excel files with the defined clinical queries and images of the Shrine criteria and the extracted data, so that to make sure that our analysts and our clinicians eventually who are using this will be able to understand how those queries were put into Shrine, so that we make sure that we're all speaking the same language. Again, when we are running this in the ENACT network through Shrine, each of the queries is run locally, in its own EHR database with independent execution. So we're not, and there's no cross-site patient data sharing, and the identical query version is distributed to each site to rerun. We come back with aggregated data. Typically, that has been with the site return in plus or minus 10 patients. We'll talk about a change that has been recently implemented more recently. We get count tables only, never row level data, so we don't ever see the exact patient level data. We just get the aggregated counts. Our floor had been 10, with sites that had fewer than 10 patients were suppressed. And then sites that gave back only had plus or minus 10 patients, 0 to 10 patients were excluded from the pooled analysis. We couldn't be sure whether those had 0 patients or not. So then the fourth step is run by our statistical team. They get the analyst email that says everything is ready. And then our statistical team will create statistical analysis from the Shrine data. And with that statistical analysis, they then create an initial report that they send back to our clinicians. And then they alert our clinicians that the report is ready to be looked at. So again, a lot of times these site level counts, they're using a random effects model analysis. So basically assuming that each site might have different things that may affect it. And so instead of that's the pooling method that we're currently using, they're using an incidence rate ratio with 90% confidence intervals to measure that effect. And then they're generating a forest plot to show how much each site is basically contributing to that pooled estimates. And so that gives you an idea and will give the clinician who's going to be reading the report back an idea whether this is seen at multiple sites the same way, or if you're having a lot of divergence between sites, or if you're having one main site that is really pulling all of the data one way. And then we have the sensitivity analysis that we're excluding the low count sites. So the sites that are under that obfuscation threshold, we are taking those out to make sure that those aren't really affecting the data. And then our final portion is that clinician report. So once we have the data back from the statistical team, our clinicians take a look at it again and come up with a structured one-page summary that they are delivered to their requesting clinician. Make sure that it's in language that the clinician will understand. And then they also are looking at what caveats they need to put into that report, as well as the statistical. There's 3 parts to the report. One is here's how we took your clinical question and developed it into a bunch of queries to put into Shrine. Here's the actual data that we got back in the statistical analysis. And then here are the caveats and limitations that we've found with our data to help you understand what we've thought about, what we haven't been able to utilize, and coming up with the data that we're giving back to you. And then we deliver that finalized report then on CCES clinician. With that clinician, again, has a one-page summary that includes the cohort description and the primary results, plain language interpretation for the clinician to use, the forest plot so that we can kind of see how all those contributing sites are contributing to that the data that's given back to them. And you have the caveats and limitations. So we'll talk about any confounding that was potentially there, any missing data, any generalizability concerns, and it's those are all flagged in each individual report. So the other thing we've been using to help us with this multi-step process is we've been using the Microsoft Planner integration. So we kind of been putting each question into Microsoft Planner so we can see where things are, what are the to-dos on each clinical question, how is it coming along in progress, where that is, what's in where those each question is along the line, and then we also can see what we've completed as well. So this is just this is not the actual Microsoft Planner sheet of our in-process reports, but just a kind of an example of what it would look like. So the next thing I want to do is we have two examples I want to take you through so you can kind of see what we've done and taken from a question to the report that gets back to the clinician. So this is our first example where we had patients who were on inhaled corticosteroids and then developed Candida esophagitis. We looked at what is the likelihood of recurrent Candida esophagitis in those patients who were restarted on inhaled corticosteroids. So basically looking at what's the risk of developing again Candida esophagitis if you've had it once, if you've been on inhaled corticosteroids and then were continued on that. So basically we took a look at 4 patients who were on the inhaled corticosteroids and had clinical Candida esophagitis, and then who were put back on inhaled corticosteroids. What percentage of those patients developed it again, and compare that to a group of patients who were on inhaled corticosteroids, got Candida esophagitis, and then were never put back on inhaled corticosteroids again. How often did they develop Candida esophagitis? And so, as you can see, those were the counts for the patients of the intervention group and the comparator group. In the first column, the patients with the recurrence. In the second column, and the incidence per 1,000 patients in the third column, and then our accrued incidence rate ratio in the last column. So again, looking at this data, it looked like if you were put back on inhaled corticosteroids after you had an initial Candida esophagitis event, that you are 1.4 times more likely to develop recurrent Candida esophagitis if you were put back on your inhaled corticosteroids. And so that's the basic results that we would be able to give back to a clinician and for them to be able to take back to the patients. But then in interpretation, we talk about what we talk about, what do we think about, what were we not able to analyze as part of this. And again, the clinical query there was, did I assess the benefit of the patient to the inhaled corticosteroids? So we didn't look to see like, hey, are these patients with COPD, did they have less hospitalizations or less COPD exacerbations? We weren't able to look at that. So we didn't see the benefit, weren't able to address the benefit of the inhaled corticosteroids. And then we couldn't ensure that the exposure control groups had the same rates of other risk factors, such as HIV, were they on other immunosuppressants, did they get, were they exposed to chemotherapy, did they have diabetes? So we weren't able to look at those things as well. So when the clinician is able to take this report and take it back to the patient, they can understand kind of what are the things they were able to look at and what things they weren't able to look at with this, and be able to take this back to the patient. So that's one example of a report that we created using this method. Another one is we had a question that was looking at SGLT2 or GLP-1, so 2 of the newer classes of diabetes medications. We looked at those and compared them to other anti-diabetics on whether or not those patients who were placed in either of those groups were able to, whether the SGLT2s or GLP-1s were able to reduce the risk of limb loss compared to other patients with on other diabetic medications with peripheral arterial disease. And so they were asked us that question. Then they also asked us the question of, is there a minimum exposure window to which this effect may appear? So one, we were able to say, at this point, we're not able to give you the minimal exposure period when those two groups might have some divergence in the lines of rates of lower limb amputation. So we weren't able to answer that part of the question, but we were able to answer the general question or get some evidence for the data for the general question. So we looked at all the patients who were on the SLG2 or GLP-1 inhibitors, which was about 67,000, or other diabetic meds, which is 209,000 patients roughly. We looked at the groups who had a lower limb amputation in each of those groups, which was 3,600 in the SLG2 or GLP-1 group and 17,000 in the other group. Looked at the crude incidence per 1,000 individuals, and we could see that was 54 and 80. And the crude incidence rate ratio, which looked to be that our patients who were on SLGT2s or GLP-1s had a lower incidence of lower limb amputation. Again, we showed the interpretation of what that means. Let me talk about the limitations. That one, we didn't take into consideration that patients may be taking medications from both groups, especially in the SLDT2 and GLP-1 group, and they weren't just taking those diabetic medications. We weren't able to look at the duration of treatment, the dose of those medications, what the baseline characteristics of the cohorts and the comorbid conditions. And we also were able to only use single ingredient medications in our analysis. So those are kind of what we saw as the data we were able to give back for this clinical question. And then in this one, we ran a forest plot. And that is shown here. So here you're able to see all the 17 sites that when we ran this and ran all the queries in ENACT that we're able to get data back on, and who gave us data on all of the queries. And so we were able to look at the incidence ratio for each of those sites. And so here you're able to see that, while we did have one site 4, that kind of showed the opposite answer, the vast majority of all the sites were right around the answer that we were getting. And so it wasn't one site in particular that was driving all of the data for this question. I think this is also helpful in looking at when we look at the clinicians are able to look at the data and be able to feel more confident in the data that we're giving them. So next, let's talk about our current limitations and challenges that we're experiencing. So one thing that we've been experiencing were some limitations with the Shrine portal itself. Another one was getting questions from clinicians and marketing, and the other was turnaround time. And I'll kind of talk about each of those in detail here in a second. For our Shrine limitations, one, we had some obfuscation limitations that have been more challenging. The previous obfuscation limitations were the plus or minus 10. We talked about and I'll talk about in a second what's been going on to address that. There was also previously, when we first started the project, there's only one sequential event logic that you could have within the Shrine portal, which really limited how many sequential events that you could ask or put into each query, and really made us rely a lot on having to use dates to create sequential logic, and which are not as good as being able to have just sequential logic itself within the Shrine portal. For our clinician question acquisition, one of the things was how are we able to make it easier how easy was it for clinicians to be able to submit a question? And then also, how do we get it to be top of mind for clinicians? Because one of the big barriers that we are experiencing is clinicians have got to think about the service to be able to submit a question. If they don't think about the service, they're not going to submit a question. So we got to be able to figure out how do we get them to hey, this is something that's out there. This is something that's potentially useful, and I want to figure out a way to get them to be able to think about this is something I want to utilize. And then also we need to think about how do we roll out to other sites other than just a UCSD, because our initial rollout was just to UCSD, and we kind of had a slow rollout there, thinking about where we'd roll out kind of a more targeted rollout with a more generalized rollout that came more recently. Thirdly, we want to think about our turnaround time, because one, right, our current workflow uses a lot of clinician time, and those clinicians also are very busy clinically or doing other research as well. And so when they get a question, they may not be all be available right at that moment to do all the work to generate to take those clinical questions and turn them into clinical queries, and also help look at the final report right at the very end. And so how can we help reduce that clinician availability limitation is one of the things we've been thinking about. And then also, is there any way to help with this multi-step process? Because this process also has lots of different steps to it. How do we make sure that that runs smoothly? So looking at the Shrine limitations, the action plan, we've looked at that. I know that many of you are probably aware that with that we've been able to get the ENACT network to change the obfuscation limits to move from ±10 to ±3 patients per site. Sites are slowly adopting that. So if we go into Shrine now, you'll see a few sites that have when you run a query, you'll be able to see which sites have already adopted this new obfuscation limit versus which ones are still on the old obfuscation limits. And so as more and more sites adopt the new obfuscation limit of ±3, that will definitely help with reduces uncertainty with smaller populations. So it will increase the number of questions we're able to answer and not have them just be questions where we have tons and tons of patients, but be able to answer more of those questions with more limited populations. Then also, and in Shrine, they've more recently added a second sequential event, which is really going to help us be able to think about, again, increase the universe of questions so we're able to answer more confidently with having two sequential events within the Shrine logic. But now the question is, do we need a third event, or if we add a third event, how would that affect how Shrine works potentially? And then next, for the clinician question acquisition, we're working on making it easier for clinicians to submit questions. Right now, we've been using an ENACT CCES email at UCSD. We're working on creating a new online submission portal that we can put on the ENACT website, so it'll help with our rollout to other sites that we won't have a UCSD specific portal of entry for submitting questions. And we'll also have examples of clinical questions that have been successfully analyzed on the portal to help clinicians understand, hey, this is my question, like one of these questions potentially, and help people think of questions as they're looking at that submission portal. And then once we have that submission portal on the ENACT site, we can go and potentially create, if we need to, we can create portals at each location where we have the CCS rolled out to, and make a local entry that redirects to that one submission portal on the ENACT website. Again, to help be top of mind for clinicians, we want to continue our advertising. I think this as we see that it'll potentially need continuous advertising until we really have a lot of uptake, and then I'm hoping then we'll get some more word of mouth advertising, which I think is when we know we've really taken off. And then really the sharing of successful questions through events like this, through events locally as well, and showing how this might benefit folks. And then finally, roll out to other sites. We are currently live at UCSD. We're trying to roll out to Pittsburgh right now, maybe potentially looking at UT Southwestern as the next potential target. But then we could have potentially all of the different sites that are in the ENACT network to be our potential sites to be rolled out to at some point in the future. Finally, I'm talking about our turnaround time action plan. We're looking for limits on clinician availability. We're looking at AI to take some of the clinical questions from taking the clinical questions to the Shrine queries. We've partnered with the ENACT NLP group so I think this partnership, it looks to be much more promising than using our initial foray into this and using publicly available LLMs, because I think we are able to put the ENACT the Shrine ontology into the workflow and have much better response with a co-developed AI workflow. And I'll show you that in a screenshot of the beginnings of that on the next slide. And then finally, we have a multi-step process that we're working on. We're using Microsoft's Planner, as I've shown. We're working on that to try to streamline communication workflows to make sure that as soon as one area has finished their work, we can pass it on to the next area uh, to help them take the baton. And then finally, we're developing some pre-designed statistical analysis for certain query structures so that as soon as the statistical group has their data, they're able just to put it into a model that they've already pre-designed and then to have quick turnaround on this in the statistical analysis. Finally, here's the screenshot of the AI integration with the ENACT NLP working group. We have a preliminary version of the tool that's built and currently undergoing validation testing. This tool will have basically take us from, it doesn't have all the steps on there, but basically taking everything from the time when the clinician submits that initial question, does the initial literature review, helps with the ontology mapping, and goes all the way to the point where we're able to look at have the clinical queries ready. But as this workflow is been created, we have, while it allows for automation, there are clinician in the loop steps within this to help make sure that that clinicians are able to utilize this, but have ways to be able to step in and say, hey, we need to add these couple other uh, um, uh, words or definitions when you're thinking about developing the queries, or making sure that this is the queries that are designed to answer the question appropriately. So there's definitely clinician in the loop to make sure that the automated AI tool that we're using does not go off the rails and provide an incorrect queries that'll be analyzed. And with that, I will open it up to questions.
Thanks very much, John. So if people have questions, if they want to type them into the chat box or maybe raise your hand. One thing I just wanted to point out is, for example, that candida esophagitis question. The idea is to engage patients in their own decision making. So we can now define with CCES what the relative risk of restarting the inhaled corticosteroids might be. But it really is up to the patient and their doctor to decide whether it's worth it. If their inhaled steroids are really important for their comfort of life, the overall rate of having a second episode of esophagitis is pretty low. And so maybe someone will decide to go back on it because the risk is low and it can always be treated again if it recurs. So that's where we think the patient participating in their own care would potentially benefit. John, if you look in the chat box there.
Yeah, I see there's a question there. So not sure if I missed this, but is it possible to have software run adjusted analyses for the IRR? For example, we can set a list of variables to have it adjust like age, sex, race, ethnicity, et cetera, in the query. It might be nice to have both crude and adjusted estimates. I'm not sure there's anybody from the statistical team on here because I'm not on the statistical team there. Um,
I think it will end up being dependent on the type of analysis. So the numbers are very small for the Candida esophagitis example, but for the SGLT2 and GLP, there were thousands and thousands of patients. So then you have the opportunity to start to stratify based on demographics and other influences.
Yeah. For the amputation study, any worry about the timing of the lower limb amputation? Could it occur before the GLP? So we had put them as that. We'd use that as the lower limb amputation to occur after the. As far as we know, that for the group that had the GLP-1, we looked at ones afterwards. So we can't just say that, hey, if you have a patient who's already had a lower limb amputation, can the GLP-1 prevent another lower limb amputation? But we looked at for ones afterwards. That's one thing we weren't able to say as far as we because of the number of 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 the events when we were looking at it, we only have one, and so we only look at basically saying, hey, we have the GLP-1, and then you have a lower limb amputation afterwards. We weren't able to look at whether you had that before or not, and so we could go back and analyze it that way. But I think you're gonna have a very small number of folks who have the lower limb amputation before they're put on the GLP-1 uh, likelihood. So. But I understand that is a concern about that, and so that's why we kind of put there was some we couldn't put all the timing of the events as a caveat. But yes, great question,
So there is a question from Malar.
Millar.
Uh, yeah, this is for the question from Catherine regarding how to do an adjusted, both crude and adjusted. For this project, we just did a meta-analysis, which is an unadjusted estimates. We just created IRR without adjustment. But I also did a study for comparing SGLT2 with other two classes of drugs where we did a adjustment for age, race, sex, and ethnicity using binomial regression, which is a different study. So for this quick turnout of events, we did not do any adjustment for other covariables.
All right. Any other questions or comments?
So the turnaround times are on the order of a week or two currently. We're working on trying to get it down to a day or two with uh, uh, the natural language processing teams, AI assistants, because one of the things is we're seeing with relying on clinician availability is just you're trying to have a small group of clinicians who are on the project time, making sure that they are available when the question comes in. Is one of those things that is one of the more challenging things. And if they're having to spend multiple hours, defining the question and the query, is that just finding the time that they're able to get to it, whether they're able to get to it right away or later on in the week, per se. And I think the AI tool will help really reduce that time and help make those turnaround times get to be our goal is to be a day or two.
Yeah, but it's a great question and it comes up all the time. So what this is not designed to do is, for example, in an inpatient setting, somebody has an arrhythmia and you ask, what's the best antiarrhythmic to give this patient now and expect the answer to come back very quickly. Most of this is outpatient, and in most cases, you would have the luxury of having the query come up during a patient visit, then submitting the query afterwards, and then at the next visit be able to address that with the patient. Perfection is kind of the enemy of the good here. Our goal is 48 to 72 hours right now. It takes a little bit longer than that for the reasons John mentioned. Um, but the notion that it would be a turnaround time that would be fast enough to answer a question within a patient visit is probably unlikely for the near future. I think even with the use of AI, I think it would be very, very difficult to do that.
Yeah, I think you'd have to have almost AI with no clinician in the loop to be able to do that. So we are not there at this point.
And yeah, thank you. Thank you very much, everybody. Really appreciate your attention and happy to answer questions later on offline. Either John or I or Elena would just direct the question to one of us, and we'll try to get back to you.